On today's episode, we're going to be talking about genetics and sharks and how to detect specific sharks that have been subject to the fin trade, or a turtle that may have been illegally taken and it has to be put back in the right spot. We're going to be talking to Diego Cardenosa, who is a postdoc at the Florida International University in Florida, and he is going to talk about his work with genetics and sharks and all these different types of animals. It's a fascinating conversation. So let's start the show. Hey everybody, welcome back to another exciting episode of the Beyond Jaws podcast. I'm your co-host Andrew Lewin, here with my co-host Dr. David Ebert. Dave, are you ready to talk about genetics and sharks?
Oh yeah, absolutely, Andrew. I'm really excited about this particular show with Diego. I've kind of known him a little bit over the years, and I've been really excited to have him come on and talk about all of the work he's doing. It's fascinating stuff. Talks about what it's like to work in different countries, particularly in Hong Kong, and getting around. And I think his whole story about working for Interpol and everything is just amazing. It's not something we get every day when we do one of these
Absolutely.
episodes. But it's a fascinating story he has, and talks about, of course, what I really wanted to hear about was the hammerhead shark. So I'm really looking forward to learning more about these little hammerhead sharks that he's found off the Pacific coast of Ecuador, which is a fascinating story to these two critically endangered species. So, so I'm really looking forward to this one.
And they're adorable looking sharks, and so I can't wait to talk about them. They're very cute. All right, let's get into the interview. Here is Diego Cardenosa talking about his work with genetics and sharks. Enjoy the interview, and we'll talk to you after. Hey, Diego, welcome to the Beyond Jaws podcast. Are you ready to talk about sharks?
Yeah, well, I'm always ready to talk about sharks. Thank you for having me here.
Well, welcome to another episode of the Beyond Jaws podcast, where we have an outstanding early career researcher. We have Dr. Diego Cardenosa, who's a distinguished postdoctoral research fellow at Florida International University. Diego's research interests are focused on determining the species composition of the international shark trade, the identification of geographical regions of the origin of these wildlife products, and supply chains at the highest risk of illicit trade, and the development and implementation of rapid, portable, and inexpensive import DNA protocols to enhance the detection of illicit wildlife trade by law enforcement in major wildlife trade hubs around the world. In recent years, he's even worked with Interpol and law enforcement agencies in several countries, such as Hong Kong, Peru, Ecuador, combating trafficking of sharks and rays, anadromous eels, and freshwater turtles, while also conducting the largest shark fin market survey in the world. In addition, he works closely with countries in South America to reduce shark bycatch and create long-term programs to assess the status of shark and ray populations in the region using telemetry, environmental DNA, and beta remote underwater video approaches. He's received numerous awards, including the Blue Marine Foundation's Innovative Ocean Award in 2024, the Explorers Club 50 People Changing the World in 2023, the Directorate of Criminal Investigation and Interpol Medal 2022, the Florida International University Distinguished Postdoctoral Research Fellowship, and the American Elasmobranch Society's Samuel Gruber Award. Boy, and that's just some of the awards he's won. And among his...
That's my favorite one.
That's his favorite one. Of course, it has to be.
The Gruber Award.
And and he did his academic background is he did a bachelor's of science and a master's of science at the Universidad de los Andes, and he did his PhD at Stony Brook University in New York. And with that, welcome Diego to the show.
Yeah, thank you.
Yeah. So our first question we always want to start off with our guests is, how did you get interested in sharks and in marine biology?
That's a question that comes a lot when I talk about sharks, and the funny thing is that I don't remember. It's one of those things that there's always been a shark in my head since I can remember. And I remember my mom actually has drawings when I was like 3 years old of like a hybrid orca and kind of shark picture. So nobody knows really where it came from, but it's always been there for some reason. And I wanted to be a shark biologist while my friends wanted to be firefighters and police,
Right.
Yeah, and it turned into a career, and I guess my parents always supported that idea. So
growing up, you grew up in Colombia, is that correct?
yeah, and you might think, oh yeah, tropical nation, whatever. I grew up very high up in the mountains, like 2,600 meters above the sea level. So the ocean was always super far from me. But I always kept on reading about sharks and watching documentaries and all these things. So always kept, an interest
Yeah.
about it.
So that's interesting. Yeah.
Did did your family go to the beach very often when you were growing up? Did you,
Not very
did you,
often,
did
but
you
when we had holidays from school or anything like that,
mhm.
I never went. I've never been to Disneyland or anything like that. All our holidays were to the either Colombian Pacific coast or the Colombian
Mhm.
Caribbean coast,
Right.
and yeah, to watch like the whale shark, the humpback whale migration, or the whale shark
Mhm.
migration, or yeah, things like that. So we always went to nature holidays. So that always kept me
Interesting.
like
Mhm.
fueled up with
So
nature and the ocean.
So,
were your parents very nature focused then? Because if they wanted to go look at nature on vacation.
Yeah, yeah, they loved it, and they were scuba divers way before I developed an interest in this.
Wow.
So
oh wow.
obviously, when they came back from their trips, they were, oh, we saw a whale shark, and they were telling about these stories and these things, and yeah, it always fascinated me to hear their stories, and I always bugged them to get me to scuba as soon as possible. So I did my first course when I was like 11 years old. Yeah.
Wow.
Nice.
Jeez,
Nice.
that's young. I don't even know what's the minimum age you have to be. That seems
I think
super
it's
young to
10
be
I
now.
think
scuba
it's
diving.
between 10 and
Is
12,
it
yeah.
10?
For Patty at least. For Patty at
Wow.
least is 10. Yeah.
Wow.
Yeah.
That I know it was not that young. Of course, I'm a lot older than you guys, but I know it was not that young when I was coming up. So that's amazing.
It
You get
used to
diving that.
be
Yeah.
12. It used to be
Mm-hmm.
12 and then it was 10. I found out recently it
Oh.
was 10.
Yeah.
Oh, okay. Okay.
Yeah. Pretty cool to be an 11-year-old breathing
So
underwater,
so
$100.
chasing
so
sharks and chasing rays.
I so when you go, as you kind of went on, like say to high school and stuff, and you go see like a guidance counselor, or to go to like particularly in high school, and they ask you like what you're going to do. Did you tell them like you're going to go study sharks?
Yeah, always, always.
Yeah,
And now that I developed this into a career, I had a lot of friends and people from, the school bus
hmm
that wrote to me on Instagram or Facebook, and they told me, "Oh my God, you end up being a shark scientist. You always talk about sharks in the school bus."
yeah,
So, so yeah, I think it was not a shocker for anybody that, oh yeah, the guy ended up being a shark scientist.
yeah.
That's
I had to
amazing.
ask that. Yeah, yeah. A lot of people you get into high school and they're college is one thing when you kind of say that because at least you're in college at that point. But when you're kind of like in middle school or high school and you're telling them you're going to go to sharks,
Hmm.
the reaction from the guidance counselors is, okay, that sounds,
Yeah,
I guess a plan.
exactly.
so,
Yeah, like I
so
want to be
you
an
obviously, so I
astronaut.
was,
Yeah, sure.
yeah, yeah, exactly. Well, Enrique Cortez, you must know Enrique. He did become an astronaut for Spain. Funny
Oh
enough,
wow,
interesting
Yes,
enough.
he
Yeah, I don't
did.
know
I didn't know
yeah,
that.
he was one of like, was it 5 or 7 astronauts in the Spanish space program? He was
Wow,
one of the ones selected. And then he ended up giving up the space program to go study sharks. So it
well,
was that
good
was
for
kind of
him.
a so
There's
it's
no
not a
there's no
completely
sharks
far
out
fetched.
there, so
No, space sharks up there. Of course, it would be a title of an interesting movie. One of these shark attack movies, sharks from space, or I guess they have something Sharknado. It's
Well, there's
so
Sharknado, but there's also Shark. Oh, was it Shark with lasers? There's actually a movie where they come from outer space and they land in they're like alien
is there?
species. It's a
I,
very old movie.
that
A
one missed
friend
me.
of
I
mine
didn't
showed
know
it.
that.
Oh,
Yeah, a friend of mine showed it to me
I
on
didn't know
at
that
a
one either.
cottage, like old VHS tape, and it's like from
Wow.
the 80s, and
Oh
the
my
the
God.
special effects is exactly what you would expect from the 80s.
Yeah,
It's awesome.
yeah, yeah. So Diego, when you get through high school and stuff, and you just got this dream for going for sharks, and you went to the universities you picked to go to. Do they have a particularly program, a good program in marine biology when you went to in
No, I, it
Columbia?
was actually
Uh,
it was actually not a marine biology course. It was a biology course, and I
Okay.
felt like
okay.
I, I met a scientist, a Colombian scientist back in the day, and he knew that I was very passionate about sharks, but I guess his advice, and I took it, was why don't you use it or think about it as a funnel. Why don't you start wide, learn
Right.
about all these different things, and then you start narrow it down as you go through.
Yeah.
I thought it was a good advice, so I started with biology. Very little marine stuff really in biology. To this date, I never took a shark course. I've never been on a shark on a, I've given it, but
Hmm.
I've never been the student of a shark course. So it's always
Interesting.
myself just studying and reading about them. And then and then when I was about to finish, almost a year before, I was at home on just a normal weekend, and I saw on Nat Geo or Animal Planet. I saw Dr. Gruber on it,
Mm-hmm.
and he was talking about his field research station in Bimini and all these things. So I started looking on the internet about it, and I realized they had this volunteer program there. So I ran to my dad's room and I told him, I found what I want to do next semester. I need you to trust me on this one. Let's postpone that semester, just not go to university for that semester. And I want to spend that semester at Dr. Gruber's field station. And he
Mm.
was like, oh, this sounds crazy. But I was like, trust me on this one. This is the right move. And I
Mm.
applied and I ended up staying there for like 5 months.
Wow.
Wow. Now, what
Wow.
year were you in for your undergrad when you decided to pause it for a semester?
I was I had a 1 year left, I think.
Okay, so you were
Okay.
almost done.
Almost done.
Yeah.
And then while I was there, I met Tristan Gottridge and Steve Kessel and Kristen Stump.
Right.
Hmm.
That was like that time where all of them were the PhD students at the station.
Right.
So I helped them with all
Hmm.
their research. I ended up convincing Christine Stump to give me a set of her dissertation so I can do my undergrad on it. I came back to Colombia, convinced us to a professor of the university in Colombia to sign off on it and like take me
Yeah.
under her wing and her lab to finish this project. And then I went back to Bimini to collect more data on the PTAG, on the long-term survey that they did with the mark recapture data on the
Mm-hmm.
nursery areas of the
Hmm.
lemon sharks. Um, and then with that same professor in Los Andes in Colombia, we she took me on to the genetics route. I almost failed genetics as an undergrad. I hated it. Um, yeah, I graduated from undergrad not wanting to do lab work. I wanted to be a psychologist and
Right.
you know,
Hmm.
be in the field and things like that. And then she took me under her wing and then kind of like showed me how genetics could be applied to for real powerful things in
Mm-hmm.
conservation. I feel like when I was an undergrad, nobody really explained me why or how could we use it
Right.
to solve some of the pressing issues, at least for sharks that were in my interest. And then she showed me and here we are. The rest is history.
Yeah. Here we are. Yeah. So you got through your so was your master's degree on worked on sharks then? Did it include
Yes, yes. So
master's thesis? Yeah.
I ended up doing the genetic population structure of the pelagic thresher in the Pacific Ocean.
Oh,
It was a pretty
okay.
cool
Okay.
idea because when we started it, we partnered up with Dr. John Hyde at the Southwestern Fisheries Center in La Jolla.
Mm-hmm.
Mm-hmm.
And he had samples from
Yep.
Taiwan, from Hawaii, from everywhere else, and we had samples from
Mm-hmm.
Colombia and Ecuador. And when we put the data set together for microsatellites and mitochondrial genetic markers, we realized that even though you grab a pelagic thresher in Ecuador or one in Taiwan, if you see it, you look at them, they're the same. The pelagic threshers, they look the same, nothing in particular distinguishes them. But when you look into the genetics, both mitochondrial and nuclear, they are completely different. They've been separated for
Mm-hmm.
millions of years. There's no contemporary gene flow. So when we saw that data, it was a little bit like I was blown
Yeah.
away by it, and yeah, we ended up
Mm-hmm.
publishing that paper, and that was kind of like my first approach with genetics.
Okay. Okay. Oh, neat, neat, neat. So you get through your master's degree. Then how did you happen to pick Stony Brook for your PhD?
That was, I think, like going back in, like thinking backwards, I think that was one of the turning points for my career. So Dr. Damien Chapman was the external advisor for my master's student. I knew Damien from my Bimini days,
Oh,
actually.
okay.
So I knew
Oh,
him for
okay.
a long time ago.
Oh, okay.
Um,
Okay.
and I told him, hey, you
Yeah.
know what? I want to do a PhD with you. And he was like, yeah, I want you to do a PhD with me. So I was like, okay, that's
Awesome.
awesome. Yeah, perfect scenario.
Yeah.
And then when we started talking, he mentioned that he had um, projects in Belize that were with the local fishermen, very field oriented, and kind of like where I thought I wanted to take my career. So I was like, oh
Hmm.
yeah, that sounds like an interesting project. And he was like, I also have another one in Hong Kong. And I was like, oh, tell me more about it. So when he mentioned that they were starting to do the surveys in the markets and these things, I realized that it was a lot of heavy work in the lab, not as much as in the field with the sharks. But I also realized that since Shelley Clark, nobody has ever done anything on the fin trade. So there was a
Hmm.
major scientific gap there. So it got me thinking, and I was like, okay, yeah, I can do the ecology side, which is, the fun part and being in Belize and all that, or
Yeah.
I can become a lab rat, um, but potentially turning things, for like having a lot of like groundbreaking science coming out of that. So I took on that one, and yeah,
Hmm.
I think it was one of the best decisions I ever made.
Well, that's a big decision to
Oh,
do, not go in the field. I mean, growing up, always being like, I want to be with sharks. I want to, learn about them, learn about the behaviors, learn about their ecology.
Yeah.
That, especially as a young graduate student, that's a big decision to make, to be like, no, I'm going to be in lab, but I'm going to pick something that could have some pretty groundbreaking, research that comes out of it that could help the conservation trade. There's still that, like, I don't know if it's selfish or not selfish, but there's still that need to go into the field and that want to go into the field. What was that reason that you're like, no, I'm gonna do this, because even though I'm in the lab so much, like, was it the conservation outcome that could come out of it? Like, did you foresee that happening,
Yeah.
or did you question that at all?
No, no, no. The project was so well um set up,
Yeah.
and as I said, like I've always been reading all the science that come, well, not all the science, but a lot of the science that comes out, and trying to keep track of what people were doing. And yeah, as I said, like since Shelley Clark, there was very little information on the shark fin trade, which was one of the largest conservation problems out there. And there we had at least 10 years of no like major papers on those markets.
Right.
So I quickly
hmm.
identified that was something that I would enter a niche, uh, a niche area of the conservation of sharks that needed somebody dedicated on it. So I never looked back after that.
Nice.
How did it work? How did it work when you went? Did you work with people in Hong Kong to get around? Obviously, going to the markets. Unless you speak Mandarin, we just didn't pick up on that in your somewhere on
No,
your CV.
um, no, so yeah, we have collaborators in Hong Kong. One of our largest collaborations is with Stan Shea from Bloom
Oh yeah,
in Hong Kong.
he's been on.
Yeah.
Yeah, he's been on our show.
Yeah, so he's
Hmm.
been in our project since the inception, and basically the project runs because of him. So he
Hmm.
goes through the markets, and he's the one that buys the samples. And when I used to go there, he was the one showing me around and making sure that I was, okay as a young Colombian in the middle of Hong Kong.
Yeah.
So he was
Hmm.
definitely a lot of help.
Okay. Yeah. Yeah. I mean, I had a similar experience when I was a PhD student. I lived in Taiwan for 3 months, going to all the markets there, and so I know how it is to try to like navigate in Chinese when you don't really speak Chinese at all. Try to learn
Yeah, the
some.
good
So
thing
it's
is in
an
Hong Kong they
interesting.
speak English very fluently. So
Yeah.
Like language wise was not a much of a problem, but also then back in 2018 I moved to Hong Kong for an entire year.
Oh,
Oh
wow.
wow.
Yeah. So before since I started in 2015, I was going like 2 or 3 times a year
Yeah.
for the collection
Hmm.
of all the data. But then in 2018, we developed this quick real-time PCR that identify all the cited listed species back then. And I moved to Hong Kong to basically teach the local authorities how to use it. And I went to inspections of large containers with them, and we ended up busting a bunch of
Hmm.
of shark fins and all that. So it was a big cultural and
Yeah.
yeah, and an academic experience, but
Oh,
more the
yeah.
cultural because I had to as a Colombian,
Hmm.
non-native in Hong Kong, I had to move there for a full year and work with the basically the Hong Kong government at a time to apply
Hmm.
this new technique to them. So it was
A lot of challenges.
a lot of challenges, but
Yeah.
yeah, they're using it and they're ramping up their inspections and they're
Yeah.
combating
Oh,
illegal
that's
trade.
great. That that
It's really
that's a
it's interesting to go.
phenomenal experience. Yeah.
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 Yeah,
Go
for
ahead.
sure. It's interesting to go to Hong Kong where this it's pretty much the home. You can almost consider it like the home of the imports of the shark fin
Yeah.
trade, right? It's a big hub for that. And to go there and to work with the government and the police on figuring out, what's contraband, what's not, it's not a very safe endeavor. Like it can be dangerous when you're going like did you ever feel like you ever in danger when going and looking at these containers or looking at these places that had shark fins? Because we've heard the stories and it can be very scary, especially for researchers who are not used to being enthralled in that and judging just based on the information that you provide us too, having working with Interpol and so forth, which we'll probably get into. You're dealing with authorities, you're dealing with police, and obviously some police activity can be dangerous. Did you ever feel that danger, or did you ever feel like worried about what the work you were doing and how it affected the quote unquote business of this illegal activity?
Right. So when we started opening the containers and all that, it didn't seem dangerous at all. We were in government facilities and
Right.
it was not in the street or anything like that. So
Gotcha.
definitely safe. Um, and I'm just the provider of the tool. I don't bust people. I don't arrest people. I'm not the judge. I'm not the. I just,
Right.
I'm just providing scientific advice actually, and the tool itself. But there was one time when I was living in Hong Kong that once I was able to convince them that the tool worked and it was efficient, fast, it told them what it needed to know. Then the authorities asked me if I could develop something similar for the European eel, which
Mm.
is all this also like a major wildlife trafficking issue. I don't know anything about eels, or at the time I didn't know a thing about eels, but the scientific principles to develop the tool were the same. They
Gotcha.
still apply. So while I was living there, I did all the work. I designed the primers. I was able to bring some samples to Hong Kong so I could work on target and on-target species. And eventually, like in November, I told them I submitted the paper. It's accepted. It's good to go. Um, I wrote to the Department of Justice of Hong Kong a letter and um, attached the paper to it. This is the scientific principle on it. They accepted it, and eventually in December on a Saturday, I think I was at home in Hong Kong, in my little tiny apartment, and they called me like at 5 p.m. telling me that I needed to grab the real-time PCR machine, which was really small, and run to the airport as fast as I could because there were two smugglers of eels and they wanted to test this thing because before the technology was put in place, the only thing they could do was let the smuggler go, take on the eels, but the crime still unpunished, right? Because they didn't
Right.
have the evidence of something like wrongdoing on time. So I took the train to the airport. I arrived there, and the two smugglers were there. and
So they saw you come out.
yeah, they saw me. I came in. We opened the bags, and they were full of little plastic bags full of larvae of European eel. We ended up running the test. All of them ended up being European eel, and the two guys went to jail for it.
Wow.
So it was the first time in Hong Kong history that they were able to do a prosecution of an eel smuggler. And it was funny because one of the smugglers was Austrian, and he wrote a letter in German because he didn't speak English. And then the local authorities knew I knew German, so I had to read the letter to them and kind of like talk to the guy and try to translate what he was saying. It was a whole mess, but yeah, like I said, that was the only time that I felt like, hey, I'm kind of like helping the local authorities to
Yeah.
read the letter from the smuggler's hand. That was a little tricky, um, but I've never felt unsafe other than
Yeah.
that situation.
Yeah, for sure. And I have to ask too. You know, you're from Colombia. You live in the U.S. You live in the U.S. At the time, and then you live in Hong Kong. Where did you learn German?
Oh, I grew
How did
up.
that happen?
I grew up in German, in Bogota, but there was a German
Yep,
school there, and my dad and my mom got me to German school, so I learned
that's
German too.
hilarious.
Yeah.
That's
Oh,
so cool.
yeah, that's really it's super interesting. Is that pretty common? 'Cause I think Pancho, you know Pancho, right? As he was in Chile a little bit, he went to a German school as well, and I think so. I was kind of if one of is that fairly common in South America? A lot of the countries that
Yeah,
have
there's
German
several German
schools.
schools in Peru and Ecuador, Colombia. In Colombia, there's four.
Okay,
There's one in Lima and Arequipa in Peru. There's one in Santiago de Chile. So there's German schools in large cities in South America.
Yeah,
okay, okay. Yeah, I was kind of curious about
Argentina is
that.
heavily influenced by German language, right?
Yeah,
Is it
Yeah, they
Argentina?
have like
yeah.
historical
Yeah,
They have it.
attachments.
Yeah, sort
yeah.
of there. Yeah, yeah, yeah. So how with all this experience, how did you get hooked up with Interpol?
All this work with the wildlife forensics, and just collaborating with the local authorities. So they caught ear of what I was doing. A few years back, I presented like a new tool that is about to be submitted for publication. Um, I went to Lyon to the Interpol headquarters to present it there in a working group that they have for illegal
Yep.
wildlife trade. And yeah, one thing led to the other one, and I think that the field I entered the wildlife forensics field because of sharks, but the whole field of all these different species that are affected by it, it's super large, and there's a need for these kind of quick tools to identify illegally traded animals and parts of animals all over the world. So it's a very interesting field, and I with what I've developed for sharks, I think there's a lot of synergies and things that I can contribute to it as well.
And you also you worked on freshwater turtles.
Yeah, so in
It's,
Colombia, again, it was exactly the same as the Hong Kong example where oh, you're doing all these, quick tools to identify these things. Can you help us with this freshwater turtle? It's called the mata mata turtle. It has like a snorkel as a nose and smiley face. It's super cute.
Nice.
Um, and they smuggle them from Colombia through the Amazon into Peru, and then they're sent abroad for the pet trade. But a lot of any of those animals coming out of Colombia is illegal. But when they catch them, they're alive, and they have to know where they came from in order to put them back to location or region of origin. But if you want to do that the conventional way, it can take weeks to months. So you have to keep all those turtles alive. They might die in captivity. They get sick, and then you're releasing sick animals into the wild, which is not good. If you're also releasing one set of those turtles into the wrong population, then you're starting messing with evolutionary forces like outbreeding depression and all that. So it's delicate to know where you're reintroducing those animals, and we develop a similar tool. Again, the principles are the same. It's just you need to tweak a few things here and there. And the result was a test that within 2 hours I can tell you where the turtle came from. So they didn't need to keep
Wow.
those turtles in captivity. They just like put them back where they came from within hours.
Okay, so now now that you explain how the
Incredible.
turtles that you want to make sure they put them back in the same spots, there's obviously a high conservation concern. So that's going to lead me to an off the wall question here. So they have an issue in Colombia with hippopotamus that are in the rivers that and I didn't realize that till a few years ago when I was actually in Colombia, and I heard about all the hippopotamus down there. If people don't know, they can check it out. But it's Pablo Escobar, a famous drug dealer from the '80s, had his own wildlife reserve, and one of the things that happened was he let all the hippopotamus go into the river, which I see as a potentially catastrophic environmental disaster if those things really get out into the Amazon basin. Could you talk a little bit about the hippos?
It's I know almost as much as you do, and I agree that. So there's a big debate in Colombia about what to do with those animals. Obviously, you cannot reintroduce 80 adult hippopotamus back to Africa. It's just not an option anymore. Um, and then there's the kind of like the animal welfare camp that rightfully so think about like why are you going to execute all those animals?
Right.
But on the other side, you have to think about the local fauna that were not they're the real victims of all this. So
Yeah.
there's this debate on what to do. Should you shoot them? Should you keep them in captivity somewhere? But how are you gonna even catch all those
Hmm.
hippopotamus? Where's the money coming from? So it's a big debate. But I'm with Dave. It's an ecological disaster in the making for sure.
Yeah. I mean, I know when I was down there in Colombia, they had at an IUCN workshop. They were talking. They were talking about it. And, my first impression is just shoot them. Just they're not native, and it's going to be a disaster there. But I know there's groups down there that don't want them euthanized.
Yeah.
Yeah,
But I don't know how. I don't know, man. I mean, the habitat. Having lived in Africa, the habitat's great for those hippos. I could just see them exploding population wise. And I don't again I don't know the full extent of the area they cover, but man, I think if you don't get rid of those things, you're going to have one just
hell of a
like you just
problem.
said, an environmental
Yeah.
disaster. Yeah, hell of a
For sure.
problem
Yeah,
down there.
and it's one of
So anyway, I
those
had to bring.
typical.
Yeah.
It's one of those typical examples when there's no natural predators for them,
Yep.
so they can grow
Yeah.
unchecked.
Yeah. And they're
Yep.
yeah, and they I mean, I don't know if you're maybe aware of this, but hippos are probably the most dangerous animal in Africa. They kill more
Yeah,
people than lions, crocodiles, and anything else. I mean, they're really yeah, and people could
yeah,
check that out. But having lived in Africa, they worry about the hippos more than anything else because they kill a lot of people. And
yeah.
if you
There
get
is.
They're very
into
defensive of their territory.
yeah, yeah, and if they get in,
Yeah.
like I said, I haven't seen them in Africa. I just can I boy, I tell you, people really knew what could happen there. I think they'd be out there exterminating those things or euthanizing them. And I don't mean to sound
Yeah.
cruel about it, but it's the consequences are just going to be just unbelievable. So,
Yeah,
um,
I'm with you. Like the greater good should prevail in this scenario.
Right. Maybe
yeah.
even put them in a zoo, but I don't even know how you capture a hippo. Like,
Oh my
considering
God,
80.
those
Like,
things.
Well,
where do you
yeah,
put
if you
them?
had one, but now I don't know
Yeah.
what the
Yeah.
number of the population is, but it's large.
It's
It's
large.
not
Yeah.
a
Yeah.
Yeah,
small endeavor to take them out.
No.
no, yeah.
Yeah.
I mean,
It's a difficult
you
thing.
can't take them back to Africa. There's just not. I don't think there's enough zoos in the world to deal with
No.
them.
You'd have to almost make a hippo zoo. Like, it almost have to be a zoo of just full of
Yeah,
hippos.
but
I almost feel like.
again, hippos are big animals, and like
Oh,
to
I
go out
know.
and to try
Yeah.
to catch them alive, it's highly dangerous. That's all
Oh,
basically I
yeah.
could say it.
Yeah.
So
Yeah.
But yeah, anyway, so I had to get involved in that because that's probably something else you'll be dealing with later on is the genetics of the hippo population.
For sure. Uh,
You
But
know,
this is an interesting sort of approach in terms of, especially when you talk about the turtles, and it reminded me of a story. I think it was in Costa Rica that I covered on my other podcast, How to Protect the Ocean, where the government was worried. I think it was stingrays. They were catching a lot of stingrays and they were confiscating them at the airport before they got shipped out. But they didn't know where to. It was stingrays or some other animal. Maybe it was crabs. I think it was crabs. But they didn't know where to put them back because they're like, if you put them back in a different spot, that could have bad ecological effects, right? Consequences. Can this? You talk about the turtles. You talk about you using it for sharks as well, and shark fins. The with the type of method you're using, can you use it for pretty much any animal across the board as long as you have the genetic makeup of that animal and you're able to replicate that?
yeah, yeah. Oh, you need to have some background information before in terms
Right.
of the DNA sequence of different loci, different genes, and see where the differences are. And I guess it's on a case by case scenario. So,
Hmm.
are you wanting to detect a species? You want to detect a population, etc. Depending on what your answer is, you design the genetic reaction around different locations. But yeah, pretty much you can do it with anything that has DNA.
And as long as that species has the markers for a particular area, I guess, right? It would have to be different over those areas.
Right,
Is that
right, right,
is that
right.
right?
So,
Yeah.
if there's a species that has a large population that covers the entire Caribbean, then it
Yeah.
doesn't really matter, at least not in an
Right.
evolutionary sense. It doesn't really matter where you put them because they're part of the same breeding population or the
Hmm.
same genetic population structure. Um, so yeah, that wouldn't matter. But when there's a lot of that reintroduction into the wild of extremely endangered or threatened animals, then that's the first thing they need to know. What's the population structure in order to be able to assess what the consequences might be of outbreeding depression.
Right. Cool.
yeah,
That's such a cool
cool.
technology.
So I got to shifting towards it back to the sharks. I got to ask you about your two favorite little hammerhead sharks that you're working on there. 'Cause it seems like that's just a fascinating project. You're working with the
Yeah.
scalloped bonnet head and the scooped hammerhead shark, Sphyrna corona and Sphyrna medea. Can you? I'd love to
Yeah.
hear about that. Hear more about that project.
So when I was lucky enough that when I went to Bimini for those first times, I developed a lot of the field research methods. And although I didn't apply them to my master's and my PhD, I always kept using them, collaborating with people. But I always, in the back of my head, I always wanted to have my own field research study, and kind of like to fulfill that niche that I didn't that I wanted to do, and just move to more like the genetic side of things. But back in the day during COVID, the Colombian Navy did an expedition through. So the Colombian Navy has had a program for both the Caribbean and the Pacific coast of Colombia, where you apply to to be part of these expeditions, and they take you on Navy boats to remote locations or locations that are not super easy to access. And especially in the Pacific coast of Colombia, they're remote. There's not a lot of roads or anything to take you to those places, but they're also impacted by the internal conflict of Colombia for many decades. So it's not the easiest place to work. So when we got there with the Navy, we started working with local communities, and the idea was almost just to see what they were catching, how they were catching them, how many, kind of like try to explore a little bit the landscape and see what kind of species were part of their fisheries and all those kind of things. And then when we were retrieving the first long line that we put, I was holding the line, and then just yellow hammerhead came on the line, and I was like, damn, this. And I started trying to remember Dave's. book. What is this? What is it? I started to try to remember all these different hammerheads, and I was like, it's so yellow. It has to be Spirna corona. There's no other way. So we took genetic samples, and on the same line further down the line, a smaller hammerhead, more towards like brown, grayish, not scalloped hammerhead. I knew for a fact it was not a scalloped hammerhead. I started looking at their anal fins. I wasn't sure what it was, but in my head it could have been Spirna media. So again, we took samples and everything. I came back to the lab, ran the tests, and Spirna media and Spirna corona. Wow. I didn't know at that time locations where you can set hooks and catch these two hammerheads like that. They're critically endangered. They're super odd and hard to find. So the first thing I did when I came back from that expedition was to look for funds, apply for grants, apply for money to go back and see what I could do. So I knocked the door on Save Our Seas Foundation and the Explorers Club, and they both gave us funds. We started the project there. We were going to start where we went that first time, but then a second expedition took us further north. And when I was talking to the fishermen, I was like, "Hey, when we were back in the south, we saw these yellow hammerheads. Oh, yeah, yellow hammerhead. I can take you tomorrow to see them." I was like, "It has to be, it has to be a lie, for the lack of other word." But we went on their boats. No joking. We set the anchor. We put 3 or 4 hand lines of the boat, and 8 minutes later, boom, something was hitting the hook, and the guy was, "Let me see it. Let me see it. Yeah, this is her. This is her." Like, you mean what? This is the hammerhead. And I was like, okay. And when he pulled it out, it was a Spirnocoron on the hook. So that opened my eyes because that's the method that we needed to catch the sharks to study them, because on the long line most of them would come dead. So we moved all the project to this location, and we started tagging them with acoustic transmitters. We set an acoustic array with the help of Ocean Tracking Network, and the results of that study were published, I think around August or something last year, where we found out that they are extremely resilient. So Sphyrna corona, the location that we were studying is not, there's nothing in particular. There's not a reef, there's not a structure, it's just a mud flat. And they spend almost the entire time through an entire year within 5 square kilometers of the coastline. So that also opened our eyes, because the objective of the project was to see if maybe the sharks used an area during high tide and a different area during low tide, so we can convince the fishermen to kind of swap locations or swap areas with the sharks to reduce bycatch. That was kind of like the idea. But when we realized that they spend the whole time within those 5 square kilometers, then we started talking with the local communities, and we decided, and they agreed, that it was going to become a no-take zone. So now they are enforcing themselves and enforcing the whole community to not enter that location, because we found out that's where the sharks are. And now we're studying them with the help of the local communities to do mark recapture data to find out the size of the population that uses that location. We're also setting up in the next few months fine scale acoustic receiver arrangement to know exactly what the portion of the time during a year or more do they spend within the boundaries of that no-take zone. We're developing environmental DNA tests to see if we can find more locations or high occurrence areas through eDNA, kind of like to survey the entire coast of Colombia and see if we can find also some of the same pockets that we found so far.
They seem like, from what you're describing, they're very habitat specific. If you find the right habitat, you're likely to find them there.
Yeah, yeah, but even within the 10, like a line of 10 kilometers in the natural national park that we work, you move 8 kilometer further north than we were finding them, and there are not that many there. So it seems like for some reason there. I cannot explain it because I don't think there's anything like that in the scientific literature. So I don't know exactly why they are preferring that location. They're obviously are feeding there, and I don't know if they're reproducing there, but we've seen pregnant females and females with mating scars there. So it's definitely an important area, but we don't know exactly why, and we don't know if there are similar locations throughout the coastline, and that's kind of like where the project is going. Like, can we find these other pockets of sharks? And that's where Spirina corona. But through our fishing effort there, we've caught Spirina media, Spirina tiburo in the Pacific, so the Spirina vespertina that is in that location, and also juveniles of scalloped hammerheads. So in that small portion of the Colombian Pacific coast, you have 4 coexisting hammerheads,
Wow.
which is pretty crazy. And 3 of them are critically endangered. 1 of them is endangered. So definitely a very interesting area to study critically endangered species that they're not very common anywhere else.
Has anybody tried to do any kind of isotope analysis to kind of see how those things partition out, having like 4 small hammerheads?
Yes,
like
and Andres Navia and his research group of Squadlos Foundation did something on the stable isotope side, and we're also doing some of it just to see if there's ontogenetic shifts in their diet and or habitat use, because to this date we've never seen a juvenile. But having said that, I don't think our fishing method would catch a juvenile. Because they must be, super small. So I don't think our hooks would catch one of those little guys. But it would be interesting to see where the juveniles are. We've never seen one.
Right.
Yeah. And so when you've talked to the community, the local fishers about that, how receptive are they to like not targeting them or anything? Are they still
super receptive? It's incredible the uptake and the buy-in from the community. Obviously, one of the key concepts of the project is that we're employing them to conduct science in this location. So it's a trade-off for them. So every week we have teams of fishermen going out there and fish, and they collect the data for us, and we pay the fuel, we pay their time, we pay for everything. And they just go and collect data for us. We train them how to pit tag and read the pit tags, and they collect all this data for us. So I think that has increased the buy-in of the local communities on the project, and now they're self-enforcing it. So a few months ago, one of the fishermen from the community was seen fishing around that location, and they all went to his house. Hey, no, listen, we're in this project, and we're protecting that location, so now that's a off-limits location for nets and long lines. So it's cool to see that they're so involved that they want to keep the project and actually enforce a local MPA that is not set by the government. They set their own boundaries. They set their own ways of fishing inside or not being able to fish in there. So it's a good project by the hand of the local community, and I think that's key for the success of any project, any conservation project in locations that are very poor locations, remote locations, forgotten locations. So there's not a lot of government resources going to these areas. So through science they can see like an alternative livelihood in a different way to make money and survive in the middle of the Colombian jungle.
Yeah, that's a great story. So if anyone listening there, follow Diego on like Instagram and social media, because he posts a lot of cool pictures from that. And I learned a lot just following you on Instagram there. So I'd recommend anybody out there, if you want to learn more, follow Diego on that. It sounds like, Diego, a lot of your research has been on the Pacific coast of Colombia, but Colombia actually also borders in the Caribbean as well. Is there Do you do much on the Caribbean side at all, or has most of your focus just been on the Pacific?
No, because of this project, my focus, a lot of the research that I'm doing now, or more often, I go to the Pacific coast. But Colombia has this amazing place. We are submitting a paper on it soon, where through the Global Finprint Project, we've been setting drafts in an archipelago that is offshore in the Colombian Caribbean side. So it has 10 different
Wow.
islands and atolls. I don't know, Dave, if you heard of San Andres and Providencia Islands.
No.
No, they're like east of Nicaragua, south of Jamaica,
Oh yeah.
but they're closer
Okay.
to Nicaragua and Costa Rica and Jamaica than Colombia, but they're part of Colombian. Um,
Okay. I know what you're talking about now. Yeah. 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 Okay. I didn't
yeah,
realize
so
those were
very
part of Colombia. Okay.
yeah, so very
Okay.
remote, very isolated, lots of seamounts around. So one thing that I'm collaborating with John Carlson at NOAA is through oceanic whitetip. So we found that in San Andres, off San Andres, really close to the coast, you get a consistent number of oceanic whitetips. We Last year we tagged one and it went all the way to the south of Jamaica, but following the contour of those seamounts, and almost as soon as it was to leave the MPA, because it's a large MPA, it's called the Seaflower Biosphere Reserve, it turned around and went back to San Andres. So it's a pretty neat project that we've been doing there. I've tagged like three or four shiny white tips so far. The sad news is that all of them have been caught by fishermen, not Colombian fishermen, but a lot of these sharks then move into Costa Rican and Nicaraguan waters, and they've been getting caught there. So
I thought.
we found an area of like high vulnerability for that species in the southwestern portion of the Caribbean.
I thought Costa Rica had prohibited from oceanic white-tip sharks in their water in their
Yeah,
international waters,
but we
but
have evidence that they were caught in
Oh
their
yeah.
in
yeah,
within their waters,
yeah,
unfortunately. And
yeah.
also, so through the global finprint, we've been
Hmm.
surveying these reefs, and we actually found that these reefs have a higher relative abundance of Caribbean reef sharks than Bahamas.
Oh, oh wow.
So
Wow.
it's an amazing
Huh.
finding, and the whole region has been so basically, you know how the shark sanctuaries were established, which is the entire EEZ of a country. But Colombia has jurisdiction in both oceans, both the Pacific and Korean and Atlantic or Caribbean. So it would be hard, but the whole boundaries of the Seaflower Biosphere Reserve is 180,000 square kilometers, and shark fisheries and the same that applies for a shark sanctuary. No catching, retention, selling or purchasing of shark products applies for that region, and they've been protected there since 2008. And we can see
Huh.
that the locals are actually the super high compliance because
Right.
we see numbers that we don't see anything anywhere outside Bahamas, and in some of the atolls that we have served, it's even higher than anywhere else in Bahamas. So it's definitely like a hotspot for sharks and a successful conservation story. And if you see
Yeah.
the paper by McNeil et al. In 2020, the big global finprint project,
Yep.
those Colombian islands are showcased in their data set, and it's pretty impressive what we've
Yeah.
found.
I can imagine.
Hmm. Oh wow. Wow.
Yeah.
That's super fast. That's super fascinating. So I guess one more question. Another question here is how much time do you actually get to spend in Colombia versus your duties back at Florida International University?
No, not a lot. I travel. I would say I travel often, but I don't spend much time there. I go maybe visit
Right.
family for a day, go to the field, get it done, and then come back.
Hmm.
But yeah, I love that part of the shark science that I wanted to do in terms of field research and more involved in the water with sharks. I'm actually doing it in my own country, so
Yeah.
I'm
It's
very
That's cool.
proud of that.
it's
That's
very
cool.
special.
So yeah, so this has been super fascinating, Diego. So I got to ask one question here before we go. Is like, So what advice would you have for a young person who's wants to get into shark research or has interested in going in that path? What advice would you have for them?
Um, I guess most people would say the same, but just follow your passion. I think when you have drive, it's kind of interesting and neat what you can accomplish if you have the drive to make it work, no matter what. It hasn't been easy for me. All the challenges of being in different countries and places that I'm not, native speaking English or native speaking Mandarin or Cantonese. But somehow, if you really want it, you can make it work. And also, at least in sharks, I think it's very flashy to try to to get involved in almost the same research that has been conducted. So I could develop, for example, Florida is a heavily studied area. I could develop projects here of sharks, but it there's already very, very, very good people doing work in Florida. There's very, very good people doing work in the United States and other places. So try to find areas that are in need of your skills and that you can contribute the most to. The example with these small hammerheads is the perfect example. And also sometimes the most flashy piece of research is not the one that will have the highest contribution towards conservation, and that's what happened to me in Hong Kong. So try to the advice would be try to set the goal towards improving the conservation of sharks and not so much the fun of the research, because sometimes the work we do is more important than just the field work that sometimes is fun, but try to make your work as impactful as possible regardless of how fun it is.
Yeah, love it.
I think I like the fun. Anyway, well, this is great. This is really great,
Yeah,
Diego.
you like the
Really
fun,
enjoyed
but
having you
and I
here.
mean, at the beginning, I hated to do PCRs, and now I can spend a whole week in the
Yeah.
lab, and my girlfriend can vouch for this. But on a Sunday, I'm like, oh man, I can't wait to be Monday and run these PCRs that I've been waiting to run for. So the fun will be there anyway, if
Yeah.
you like
Yeah.
what you're doing. But yeah, try to use your skills and your drive to make the highest impact as you can.
Love it.
That,
I love it.
that's
Diego,
great.
this
That's
has
great.
been so much
Thank you
fun
so
to
much.
No,
go
no
ahead,
worries.
bro.
Thanks for having me.
Yeah,
Yeah,
no, we'll
It's
definitely
been
have you
fun.
back sometime and follow up on some more of this stuff. But really, thank you very much for coming on the show. It's really been a
No, thank you
great
guys.
episode. Thank you very
Appreciate it.
much.
Thank you, Diego, for joining us on the Beyond Jaws podcast. It was great to have you on. Looking forward to discussing more of your very interesting work on genetics and sharks. Dave, what did you think? Like this, you went all over the world, different places, working with different like government agencies from Europe to Hong Kong to like Colombia. Discovers these like two little sharks, these cute little sharks. Does work on them. Yeah, pretty cool. Like a pretty cool career so far.
Yeah.
Seems like he's just getting started.
Oh yeah, yeah. And he's a young guy. He's just getting early career researcher, and the whole story talked about how he came across those two little sharks, critically endangered. Hadn't been seen for decades, literally. And as often happens, I can tell you from experience, you go to these little out of the way villages in towns that nobody ever goes to, and some of the ones you can't really go to. Like you mentioned, you had to go with the Colombian Navy because they're not safe to go to because of drug
Yeah.
cartels and things, which is a real issue there. But they managed to found these areas that where they were unaware of them, and I thought it was even more amazing that they got buy-in from the communities there to
For
try
the
to let them
protection.
go, which is
Yeah.
amazing. That's not my experience. That's not real common that you get that. Again, I'm more familiar with that where I work in Africa, and it's like any protein, it's not going back. But
Right.
I think,
Right.
but I think the fact he found those a couple places and the communities buying into it, I think is a fascinating story. I really, really enjoyed
Yeah.
it.
Yeah, it's nice when you get that buy-in and you don't know exactly why, but you can you still get it, and it works. It works really well, and it just goes to show that like some of these fishing communities really care, and they may not be in a situation where they need the protein that badly, where they have to take a shark like that, but they're able to put it back in, understanding that there's more to be had, and more of a benefit to put them back, which I think is phenomenal. It's such an interesting career to go into genetics. You don't hear a lot about that in shark science, and we, mind you, we haven't covered a lot of it, and it's great to be able to have someone like Diego come on and talk about the impact on conservation that they've been able to have. This portable device to determine genetics very quickly, like a PCR machine. I mean, normally it would take a couple of days to get, from to getting the sample, bringing it into the lab, or shipping it off to a lab, getting it done, and then going away. He talked about the European eel when they found the two smugglers that were bringing it in. That Hong Kong anybody had never really been able to rapidly test to make sure that it is the European eel. And it was with his instrument, and they were able to prosecute based on that. And I think that's a huge movement of conservation to be able to have that rapid result, right? And I think that's the big thing about this technology, is that you can actually use it, and especially looking at using it for almost any animal, as long as you have the information you need to put in, right?
Right. Yeah. I mean, it's amazing. I mean, that's something I been around long enough in this field that I can remember decades ago they talked about, boy, it'd be nice if we had something we could use in the field to get a rapid identification on what species we're looking at. Because, especially if you're just looking at fins and they're dry and they're different states of being dried, I mean, it's really hard. You really got to know what you're talking about. I mean, I can't, for the most part, a few exceptions, it's really hard to tell what's what on those species. And as far as the individual species, but if you have something a rapid DNA thing like he talked about, man, you can figure out the species. That's phenomenal. That's a real, to me, like,
agreed.
Just haven't been around. It's an amazing breakthrough he's found there. Although I have to say, one of the things I was interested in, he didn't really have like a lot more he could add to it, but we touched on the whole hippos in Colombia.
Yes.
And I think that was, we talk about sharks and conservation and the sharks and critically endangered species like those hammerheads, but as we mentioned in the show, you have literally an environmental disaster that's going to happen if these hippos get out into the Amazon basin and proliferate, and as they're doing now. It's going to be just a disaster of beyond magnitude that people, if those things get out and get into the Amazon basin there. And so I hope they come up with some solution to the issue, because it's I know what damage the hippos can do, but they're part of the ecosystem in Africa. But boy, in South America, God, it's gonna be bad. So hopefully.
Well, they're already talking. I just looked it up. They're talking about 80 to 120 individuals. It's the largest hippo herd outside of Africa.
Right.
And as we mentioned in the show, like in the interview, it's really hard to just put them all in a zoo. There's no zoo that can hold 80 to 120 hippos. You'd almost have to have like its own sanctuary, but with borders to prevent them from moving outside. But they're very territorial, so it's a difficult situation. As you mentioned, the best thing to do maybe at the beginning was to euthanize them so they don't get released into the wild. Unfortunately, that I feel like that ship has sailed, and it's like, there's this ethical question of what do we do with these animals to protect the hippos, but also to protect the rest of the environment in Colombia, right?
I guess because they're non-native. Like, I mean, you look at non-native program. I mean, we get things like eucalyptus trees in here, which were brought in 150 years ago in California, and they want you to go out, and they have no problem going out and cutting down a eucalyptus tree and getting rid of it. Because and a number of other species. But now when you have an actual animal that people get,
like a mammal
attached, and I think people don't. With the hippos, I can talk a little bit just from my knowledge in Africa, that I don't think people really in North America and Europe really understand. These are not the cute little things you see in Disney movies. These are big animals, and they're extremely dangerous. And to try to capture them, you're really talking about potentially injuring people to try to get them. And I just, my personal feeling, like what I know of the situation, I think they just should do what they need to do and just go in there and just, because they're non-native, and just euthanize them. And just because, again, if those things get out into the bigger Amazon basin and start procreating, it's just going to be it's unimaginable
not gonna be pretty.
what's going to happen. No, it'll be bad. It'll be
Yeah, we don't know, and that's the thing with invasive species. You just don't know what's going to happen, but it's not going to have a good effect on what we see now in that area, right? So,
and honestly, I don't know what the population is in Africa, but I know I've been to places where there's no shortage of hippos around. There's
right. Yeah.
Somebody might call me on that, but I just from my own personal experience, I could see getting an email now like, "Hey Dave, they're really endangered in Africa." Well, maybe they are, but but the ones in Colombia, they're not going to transport them back to Africa. That's for sure.
Right. Exactly. Yeah.
Yeah. So anyway, so that was a whole direction I didn't expect the podcast to go in.
No. Just for you, I just looked it up. They are on the brink of getting on the endangered species list. So they're close.
Yeah.
They're not doing well,
Yeah.
let's just say, but they're getting closer to being put on the list. So, but anyway,
Yeah.
this was like I said, this is a phenomenal episode that just goes to show if you create something from a genetic point of view and a tool that could be used on a number of different animals, not just sharks and rays. It could be used for a number of different applications. It just goes to show the impact on conservation that it has. And Diego certainly has that. And I'm looking forward to having him back on the show so we can interview him about more of these special projects. So it was great to have Diego on. We'll put his Instagram handle on in the show notes so that you can get a hold of him, as well as his website. And Dave, if people want to get a hold of you, how would they do so?
Instagram is probably most active on Instagram, but also you can go to the Beyond Jaws YouTube site and leave us a message there. And the newly launched Lost Shark Guy YouTube channel with a new series Andrew helped produce, and Denise Sotomayor, who's on recently filmed, and you can see what about traveling looking for these lost sharks. Episode 1 is up, and episode 2 will be coming up here real soon. So anyway, please check that out.
Yeah.
Searching for lost sharks.
Such a fun series to produce. Yeah, it's going to be a lot of fun. Well, thank you very much, Dave, and of course, thank you to Diego. And we all of us want to thank you for the listener for listening and watching this episode. We really appreciate you doing so, and thank you so much. You've been listening to the Beyond Jaws podcast from Dave and I. We really want to thank you for doing this. We really appreciate it. And until next time, have a great day and happy conservation.